Welcome everybody. We are discussing the chapter Morphology of Flowering Plants. This is chapter 5 of class 11th biology. So in our previous session we discussed fruit and today in this session we'll discuss about the seed. So what is seed? As I said in my previous session that inside the fruit which develops from the ovary after fertilization, the seeds are present and the seeds are developed from the ovule. Okay. So, and this seed is characteristics of gymnosperms and angiosperms only. You will not find seed in bryophytes, pteridophytes and thallophytes. It is seen only in case of gymnosperm and angiosperm. But the difference is that in gymnosperm, the seeds are not protected by the fruit wall, whereas in case of angiosperm, the seeds are protected by the fruit wall. Okay. So, how can you define a seed? A seed is a dormant structure containing protective covering that is called seed coat, and also it contains the reserve food and the embryo. So why it is called dormant structure? Dormant structure means it is the inactive state. The seed is in the inactive state. The cells are living but they are metabolically not active. So there is no cell growth or there is no cell metabolism. Okay. So the seed is a living structure which contains the cells which are also living but they are metabolically not active. That's why they are neither growing nor they are dividing. Okay. And what else is present there in the seed? In the seed, the protecting seed covering is there and the seed contains reserve food and the embryo. But why the reserve food is there? Because inside the seed the embryo is there. While discussing the angiosperm, that is life cycle of angiosperm, I have told you that when the male gamete and the egg fuse, they form the zygote, then the zygote develops into the embryo. Now this embryo remains well protected inside the seed, and at the commencement of the favorable condition, when the favorable condition, what is the favorable condition for the seed? The favorable condition for seed germination is the presence of sufficient light, oxygen and water. When these three things are present in the environment, then the seed starts germinating. That means the embryo present inside the seed now starts growing, now starts developing into a new plantlet. So where from this embryo will get the food? Obviously, it is the reserve food present in the seed. So the seed has three things. One is protective covering. Second is reserve food for the embryo which will develop in the future. Then the embryo. Now let us discuss what are the seed coats. The seed coats develop from the integument of the ovule. As I have said you that the seeds are developed from the ovule after the fertilization. The ovule develops into seed. So the ovule has two integuments, means two covering. So each of these covering give rise to the seed coat. So the outer seed coat is called testa and the inner seed coat is called tegmen. Very, very important from the examination point of view. This seed coat is membranous and it is generally fused with the fruit wall. But in all cases, it is not fused. Okay. Now this seed is attached to the fruit wall. As I said that the seeds are present inside the fruit in a different arrangement. They are present as like the placentation seen in case of ovary. Okay. So how these seeds are attached to the fruit wall? By a stalk-like structure that is called funicle. And the point of attachment of the funicle to the body of mature seed is called hilum. Okay. And besides that, the seed is having a very fine pore at one end, that is called micropyle. And the role of micropyle is to permit the entry of water needed at the time of germination. So since I told you that the seed has developed from the ovule, so let me explain the structure of ovule and which part of the ovule develops into which part of the seed. Let me explain. So this is the structure of a anatropous or inverted ovule. So here you can see, and as I said that the seed is attached to the fruit wall with the help of funicle, a stalk-like structure. So here you can see this is the funicle of the ovule. So the ovule is attached to the ovary wall with the help of funicle, and the portion of the seed, mature seed, with the help of which it is attached to the stalk is called hilum. So here you can see the hilum. So hilum is present in the ovule as well as in the seed, mature seed. So I think it is clear to you, the stalk of the seed by which it is attached to the ovary wall is called funiculus, and the demarcation or the depressed part by which the mature seed is attached to the stalk is called hilum. Now come to the most important part of the ovule. That is, here you can see that there are two integuments. So two layers are there. So this is outer integument and this is inner integument. So these two integument develops into the seed coat. So the outer integument develops into the testa and the inner integument develops into the tegument. Now, what else is present in the seed? The reserve food and the embryo. So, I think the seed coat is clear from where the seed coat is formed, from the two integument. Now, from where the reserve food comes? Inside the seed, inside the ovule, now you can see that there is a mass of cell, and this cell is called, this tissue is called nucellus. So, the nucellus stores the food, and when after I have told you about double fertilization and triple fusion. So by inside the embryo sac, this is embryo sac, inside the embryo sac, the fertilization or the fusion takes place twice. That is, one of the male gamete fuses with the egg cell to produce the zygote. The zygote develops into the embryo, but the other male gamete fuses with the secondary nucleus which is present at the center, and then that becomes triploid and that develops into the endosperm. So now this endosperm, when it develops, it absorbs the nutrient from the nucellus. So who stores the reserve food in the seed? The endosperm. The endosperm stores the reserve food, and the embryo is formed inside the seed by the fusion of the egg cell and the male gamete. So this is the structure of ovule which gives rise to a seed. And I have talked to you about the micropyle. A fine pore is present at this end. Here you can see a very small pore is there. So what is the need of this pore? When the seed gets a favorable condition, that means when water is there, oxygen is there, and light is there, then the seed starts germinating. That means the embryo which is present here after fertilization, that starts germinating. So for germination, water, light, and your oxygen is required. So through this pore, all the requirements enters into the inside of the ovule, and the embryo gets all necessary requirements. Okay. Now here you can see this is the structure of the seed. So previously, in the previous slide, I have shown you the structure of ovule. See the similarity. So here you can see this is hilum. Okay. And from here the stalk is there, that is called funicle, with the help of which it is attached to the fruit wall. Then here you can see the minute pore through which the seed respires, and also it receives water. The water enters into the seed, and also the oxygen and light. Light as such, it is taken through the whole surface. Okay. So this is the seed coat, outer seed coat, and below which the inner seed coat is there. Okay. And if you open up the seed, then you can see that this is the embryo. This is the embryo, and this embryo is having a axis. When it starts developing, then it forms a axis. This is called embryonic axis, and this embryonic axis then develops the cotyledon. I will explain you what are cotyledon and how these cotyledons are formed. So here you can see the parts of seed in monocot as well as in dicot. So let me first show you the dicot seed. In the dicot seed, you can see that the endosperm. I was talking you about the endosperm. The endosperm was formed by the triple fusion, that is the secondary nucleus and the male gamete. The endosperm absorbs the nutrient from the new cellars, but once the embryo starts developing, the embryo absorbs the nutrient from the endosperm and it stores it in the form of cotyledon. So here you can see there are two white colored structure. So since it is a dicot plant or dicotyledonous seed, so there are two cotyledon and the stored reserved food material is present in the cotyledon instead of endosperm. But these cotyledon are formed by absorbing the nutrient from the endosperm. Okay. So in dicot seed, you will never find the endosperm, with few exceptions only. So they have cotyledon. The cotyledon is a part of embryo. So the embryo absorbs the nutrient from the endosperm. So the endosperm is reduced in size and the cotyledon reserves the food. Okay. Then what else you can see? Here you can see the embryo. This portion is the embryo. The whole thing is the embryo. The embryo consists of the cotyledon. Then it has an axis, as you can see here. The axis has two parts, that is one is epicotyle, other is hypocotyle. Okay. So here you can see hypo means below. Okay. And other one is epicotyle. That means the here you can see the cotyledon is present below, but the radical the radical cotyledon is occupying the above position, but the radical is occupying the below position. But here you can see the plumule, it is occupying the above position, whereas the cotyledon is occupying the below position. So it is called epicotyle. Now this epicotyle gives rise to plumule, or you can say the embryonic axis, which is present above the cotyledon is called epicotyle, which gives rise to plumule, and the part of the embryonic axis which is present below the origin of cotyledon is called hypocotyle, and that develops into the radicle. The radicle develops into the root and the plumule develops into the shoot. And besides that, you can see that the seed has the two coat, seed coat, that is outer testa and inner tegment. So this is the structure of a dicot seed. Now come to the monocot seed. On the left side, you can see the monocot seed. The monocot seeds are generally they are having the endosperm. They don't have the fleshy cotyledon. They have a single cotyledon. You can see it is present here. This single cotyledon. This is single cotyledon. This single cotyledon is often scale-like and it is called scutellum. It is called scutellum. So it has only one cotyledon which is membranous and it is called scutellum, and besides that it has a seed coat, that is testa and tegments are there. So here I have discussed the types of seeds. The seeds are of two types, that is albuminous or endospermic seed. The other one is ex-albuminous or non-endospermic seed. So as I have shown you in the here I have shown you the picture that in monocot the endosperm is there. The maximum part of the seed is occupied by the endosperm and the cotyledon is not fleshy. So such seed is called endospermic or albuminous seed. But here in case of dicot seed, you can see that there is no endosperm. The endosperm is absorbed by the embryo to form the cotyledon. So this is called non-endospermic or non-albuminous or ex-albuminous seed. I think it is clear to you. So all dicots are having non-albuminous seed, whereas all monocots are having endospermic or albuminous seed except orchids, except orchids which is having the cotyledon. So here you can see the picture of the two types of seed. So this is your the first one is endospermic seed. Here most of the portion, the dark green color structure is occupied by the endosperm, and here you can see the green color structure is occupied by the cotyledon. So cotyledon stores the food, whereas in endospermic seed the endosperm stores the food. Okay, so here is the description of dicot seed, and I have already explained you that in the dicot seed there is an embryonal axis which consists of radicle and plumule end. Radicle gives rise to root system, plumule gives rise to shoot system, and the portion of the embryonal axis between the radicle and the point of attachment of the cotyledon is called hypocotyle, whereas that is present between the plumule and the cotyledon is called epicotyle. So let me show you the picture once again. So here you can see this is the seed, this is the embryonic axis, and this slight pinkish color, that is the cotyledon. So here you can see this is the epicotyle that gives rise to plumule, and plumule then gives rise to the shoot. Then here this is hypocotyle that gives rise to radicle, and radicle gives rise to shoot. Now next, the structure of monocot seed. So I have already discussed the structure of monocot seed, but here I will tell you one thing. The monocot seeds are full of endosperm. They are endospermic seeds. They don't have two cotyledons. They have only one cotyledon, and the seeds are otherwise known as grains, and the grain is filled with endosperm, the stored food material, and that is rich in starch. Okay, and besides that, this endosperm has one or two peripheral protein layers. Very, very important, children, and that protein layer is called alluron layer. Okay, which separates the embryo from the endosperm. I will show you the picture so that it will be clear to you. So in the monocot seed or grain, the embryo and endosperm are separated. The endosperm is fleshy and rich in starch, but in between the endosperm and the embryo there is one or more protein layers are there which separates the embryo and endosperm, that is called alluron layer. Okay. And in case of monocot, the cotyledon is membranous, and that is called scutellum. And here you can see this is the picture of and this diagram often comes in your exam as 2 or 3 marks question. So here you can see that this is the structure of a grain, maize grain, and here you can see that this is the seed coat. Okay, so this is the seed coat. Then here you can see that the maximum portion is the endosperm. Here is the labeling endosperm. Then this portion is the embryo. Okay, So this endosperm is separated from the embryo by a lining, a layer of a protein, and this layer is called alluron layer. Very, very important. Now come to the embryo. In the embryo, you can see that this is the portion from where the plumule is developed, and this is the portion from where the radicle is developed. But before the development of the radicle, this is called hypocotyle and this is called epicotyle. The epicotyle is covered by a protecting covering, very, very important, children, that is called coleoptile, and hypocotyle is covered by a covering that is known as coleorhiza. So the coleorhiza is the protective covering around the radicle, and the coleoptile is the protective covering around the plumule. The plumule gives rise to shoot and the radicle gives rise to root. Okay. And besides that, here you can see that this is only one cotyledon is present, and that is membranous. It is not fleshy like the dicot seed, and this is called scutellum. Okay. So here is the comparison of monocot and dicot seed. Very clearly it is visible. Here two cotyledon, here only one cotyledon. Here you can see that endosperm is large. Here there is no endosperm, or if endosperm is present, that endosperm quantity is very less. Next, the most important event that takes place with the seed and fruits is the dispersal of the fruits and seeds. So, children, you know that the plant is static. It cannot move from one place to another place. And the place where the plant grows is always not suitable for that plant. It is not always favorable for that plant. The climatic condition, the soil quality, the light, amount of light that falls on the tree or plant, that is always not sufficient. That is not always favorable. But if the seeds are produced by the plant, and if all these seed will fall at a place near the parent plant, then what will happen? Definitely there will be competition for their basic requirement. So what are the basic requirement for the seed to germinate? I have already told you, that is oxygen, water and light. So now what will happen? When all the seed will fall at a place, then they will struggle. There will be struggle for existence, for colonization of new areas. Okay. And in order to avoid this struggle, in order to avoid this competition, there is an automatic mechanism in the plant that is called dispersal of the seed. So what is dispersal? The seeds, after it is being released from the fruit, they are carried by different agents to different parts. So what is the advantage of dispersal? First thing is that it avoids struggle for existence. Second, most important thing is that it colonizes in a area where other plants are there, so they have better adaptation. They can develop a mixed population. So these are the two advantages. Okay. And what are the agents by which the seeds can be dispersed? The most common agents are wind, water and animals. Besides that, there are certain automatic mechanism in the fruit of certain plants where explodes so rapidly that seeds are dispersed automatically. Here you can see different agents what causes the dispersal of the seed. One is by wind. You can see here the seeds are feathery like, it is very light, and often the seeds here I have shown you the coconut seed that can be carried by the water. It should be light so that it can be float on the water and it can be carried by water current from one place to another place. Then animals. Among animals, the birds play an important role especially the fruit eating birds. So when the bird eat the fruit, the seeds are often not digested by their digestive system, and along with their fecal matter, the seeds are released. The healthy seeds are released, and in this way the seed gets dispersed from one place to another place. The last diagram shows the explosion. Explosion means all of a sudden, with great effort, the fruit explodes. It bursts in such a way that the seeds are thrown away to distant place, and in this way the seed gets dispersed. So here are the some structure of the seed or the adaptation seen in the seed for dispersal. So for those seeds which are to be transported by the wind, they should be light, or they should have wing-like structure, or they should have a feather-like structure. And those seeds which need to be transported by the animals body, often you might have observed that when we move through any bushes, then some seeds or some structures sticks to our cloth. So actually these are the seeds of some or seeds or fruits of some plants which are having hook like structure, spine like structure, by which they stick to our cloth, and when we want to remove them, then they get dispersed. Then you can see that by water, mostly the aquatic plants, besides that the terrestrial plant like coconut, they also get dispersed by water. Then by bursting, that I have told you, by explosion, so rapid explosion of the fruit, also the seed may get dispersed. And most of the seed gets dispersed by human being, and especially the vegetable and fruit seeds are dispersed by human being, the edible fruit and edible vegetables. Okay. So I think you liked this video. The seed is very much clear to you. The dispersal of seed, the mechanism of dispersal of seed, its advantage, monocot and dicot seed, everything are very much clear to you. And in our next session, we are going to discuss a very important topic, that is semi-technical description of a typical flowering plant. So like chemical formula, we can also represent a plant with its floral formula. And in order to know the floral formula, we have to remember all the terminologies related to the flower. Then only we can write the floral formula after knowing the floral symbols. So in next session, we'll discuss the symbols used for different terminologies of the flower or different conditions of the flower. Then we will discuss how to write the floral formula, and from floral formula how we can define a plant or recognize a plant, or if a plant is given, if the character of the flower is given, then how we can identify to which family this plant belongs. All these things are very important, because in your for this particular year, only one family is there in your syllabus. But I don't think that this syllabus will continue. For corona pandemic, only one family is there, otherwise all the three family will be there. Moreover, this will be very essential for the NEET aspirants. So that's why in details I am going to explain this topic. Don't forget to watch this. Don't miss the session. Okay. Thank you for watching my video. Thank you once again.